friends. Today we are going to see the what are the respiratory pigments and what are the types of the respiratory pigments. So pigments are the basically protein with oxygen to form a respiratory protein and gives a particular color, so called as respiratory pigments. And it's generally constitute of two parts, that is protein and a metallic ion. So we are just going to see the what are the different types of the pigments. So here are some details. So first of all, we have to look the what is the hemocyanin. Hemocyanin is a copper containing pigments, and molecular is weight is about 1 to 7 millions, and it's a less specific, less carrying capacity of in comparison to hemoglobin. So here is the erythrocruonin, which is the iron containing metal and have a molecular weight about 2 lakhs. So next is the chlorocruonin. It's also iron containing pigments, and the molecular weight is about 3 millions. An example is polykits. Haemoerythrin and its iron containing pigment and found in the blood corpuscles of some invertebrates, help in oxygen storage. And no porphyrin pigments, as we will see the what is the porphyrin pigment which is present in the hemoglobin. So he is a Haemoerythrin. So its molecular weight is 40,000 to 1,000, 1 lakh 8,000 Dalton. An example is brachiopods, that is lingula. Myoglobin. These pigments are attached to muscles. It has some structure as hemoglobin but has only one unit, as HB contains 4 units. As we know that the hemoglobin consists of 4 units and myoglobin in comparison to that only one unit is present. It can bind and keeps oxygen, but it doesn't carry oxygen. So basically myoglobin helps to storage of the oxygen and doesn't transport the oxygen. So here's the difference between the myoglobin and the hemoglobin, and its molecular weight is 17,000. And its important thing is the skeletal muscle of diving mammals is particularly rich in the myoglobin, which will serve as a store of oxygen during a dive. It has same structure as hemoglobin but has only one unit, as we have previous discussed. Okay, here are the some other pigments like pinna hemoglobin found in the plasma of lamellibranch branch of pinna squamosa, vanglobin. It is light color, present in Asadian sea squirts, and echinochrome. And lastly, we are dealing with the hemoglobin in detail. So basically, hemoglobin has is a conjugated protein which has two parts, that is simple protein and the prosthetic group. Prosthetic groups consist of Fe metal, that is iron metal, and heme constitute of 4%, while the globin protein, which is a simple protein, constitute of 96%. It is a tetrameric protein made up of two alpha and two beta atoms, that is two dimers. And here are some details: the porphyrin ring, the hemoglobin. Here's the figure of the globin protein, that is 2 alpha and 2 beta chain, which is uh, connected by the two loosely connected salt bridge, but two beta chains never touch to each other, while alpha 1 and alpha 2 is connected to each other, loosely connected with each other by a salt bridge, that is weak bond present. Okay, is the structure of histidine side chain. Fe atoms contain of nitrogen 4. And here is the oxygen binding curve of the myoglobin and hemoglobin. Hemoglobin has a sigmoid curve, while myoglobin has a hyperbolic curve. The structure of the porphyrin ring, that is porphyrin ring plus Fe, that is iron, together form a hemoglobin. The color of hemoglobin: oxygenated from scarlet red, deoxygenated from bluish red. In lungs, Hb combines with oxygen to form oxyhemoglobin. At low pressure, Hb dissociate to form uh, dissociate into the Hp plus O2. Four pyrrole ring joined by methylene bridge are named as the porphyrin, that is four pyrrole ring connected by the methylene bridge, and in center Fe is present to form a porphyrin ring, and four pyrrole ring join to form a porphyrin. Okay, in invertebrates, HB different from the vertebrates. One is having less histidine and lysine and more arginine and cysteine. Association of HB with oxygen depend not only on availability of the oxygen but also in the pH and ionic content of the solution. HB in vertebrates is present in RBC, while it occurs in the plasma in invertebrates. As we know that the HB is in dissolved condition in plasma and also present in the RBC. So here some functioning of the working of the respiratory pigments. And The absence of nucleus in RBC in mammal allows more HB to combine. As we know that in human RBC there is no nucleus present. So in the absence of the nucleus, the RBC of mammal allows more HB to be connected within the cells, allowing for more oxygen or more CO to be transported around bone's body. If carbon monoxide, that is CO, binds with the hemoglobin, then it is irreversible process. That is permanent process. In this way, it slowly binds with all HB, and HB decreases in body, causing death of a person, so avoiding this CO gas to inhale, that is carbon monoxide, and it forms a carboxyhemoglobin. Bunsen solubility coefficient of the oxygen in blood at 37 degrees centigrade, 2.4 milliliter of oxygen per 100 ml of blood per atmosphere of oxygen pressure, called as the Bunsen solubility coefficient of oxygen. That's all about the respiratory pigments. Thanks for watching.